Hello and welcome back to viperdb. Today we're going to go over how to use a novel utility called Capsid Maps. If you have not yet seen this utility, it can be accessed from the info pages. Simply go to the desired entry, for example, 2BBV for black beetle virus. On the left side, you'll see a series of tabs, and clicking on the one titled Capsid Maps will allow you to access the utility for the current entry. As you can see, it displays all the residues found at the subunit interfaces in a single icosahedral asymmetric unit, or IAU, and indicates showing interface residues. An IAU is defined as the smaller substructure composed of one or a few subunits from which the complete capsid can be constructed. By default, the unique protein chains are displayed and can be identified by the color chart on the bottom left of the utility. On the right side, there are controls for changing the visualization. In the Show and Hide section, you can toggle on and off the individual chains, the IAU triangle, and a heat map of the residues. There are also buttons to display residues that are annotated as interface, core, or surface residues. Furthermore, in each category you can choose to display specific type of residues. For example, arginine or glutamic acid. To display other features such as residue charge, click on the corresponding button in the Color By section. Interact with the display frame as you would with Google Maps. You can zoom in or out using the mouse wheel and pan around by holding down the left mouse button to focus on specific residues. Clicking a residue will generate a pop-up displaying basic info like the name and position of the residue. This information can also be displayed by hovering the mouse over the residue and viewing the Residue Info section on the right side panel. Moreover, entering the specific residue and clicking Find Residue will allow you to locate it on the map. The S-score section at the very top stands for similarity score and allows you to compare two capsids in terms of residue location in the interface regions. Entering a second PDB ID will prompt Capsid Maps to display its IAU residues simultaneously with those for the current entry. For example, entering the PDB ID of nodamuravirus 1 NOV results in a high similarity score of 0.799, which is expected since it is from the same genus of alphanodoviruses. In contrast, the similarity score with Zika virus 5 IRE is much lower at 0.266, since the two structures are from different families. Go ahead and explore the utility. For more info on Capsid Maps, please refer to the following paper, which will be in the description at the bottom. If you have questions, send us an email at viper@scripps.edu. Thank you for using viperdb.